Good evening. I'm Mark Parcells, and I'm a professor of molecular virology in the Department of Animal and Food Sciences with a joint appointment to biological sciences. I first want to thank the organizers of this symposium for giving me this opportunity to discuss the research of the 2020 Nobel Prize in Physiology and Medicine awarded to Doctors Alter, Houghton, and Rice for the discovery of hepatitis C virus. So among the many diseases you never want to get is viral hepatitis. There are several hepatitises caused by viruses, but the most serious are the serum hepatitis viruses, hepatitis B and C. Both of these viruses can lead to complete loss of liver function, cirrhosis, and hepatocellular carcinoma, a very aggressive liver cancer. So although hepatitis is a common sign or symptom of hepatitis virus infection, hepatitis is caused by a number of viruses. So hepatitis A is a infectious hepatitis that can be transmitted by food or contaminated water. There's vaccines for this. Hepatitis B and C are transmitted in blood products through intravenous drug use, can be sexually transmitted or transmitted from an infected mother to her developing offspring. Hepatitis D or Delta virus is a viroid that is only found in conjunction with hepatitis B virus, and it requires functions conferred by that virus in order to replicate. It by itself is not a primary pathogen. And hepatitis E virus, a more recently identified hepatitis virus, is similar to A in that it's foodborne, but it's endemic in swine with spillover into human hosts. So are all these viruses in the same family? No, they're very distinct types of viruses. Hepatitis A and E fall in the family of small RNA viruses known as the picornaviridae. These viruses include polio, Coxsackie virus, and foot and mouth disease virus are all in the picornaviridae family. Hepatitis B is a fairly unique type of virus. It's a pararetroid virus. It's a small circular DNA virus that replicates via a longer than genome length RNA stage and then gets reverse transcribed back into its genome. Hepatitis C falls into the same family as yellow fever virus, as well as some other mosquito-borne viruses like West Nile, dengue, Zika. And hepatitis D, as I mentioned, is actually a viroid, a small RNA that is dependent upon hepatitis B replication for its replication and packaging. So hepatitis A or infectious hepatitis, again, primarily a food and waterborne pathogen. Worldwide, its incidence is about 1.4 million human cases per year, resulting in about 11,000 deaths. It was distinguished from serum hepatitis back in the 1940s and 1947, but the causative agent for this wasn't isolated until 1979. But since early to mid 90s, there have been vaccines for this that are delivered to children in endemic areas, as well as if you're traveling to areas with high incidences of hepatitis A, you can get vaccinated for this as well. For the serum hepatitis viruses, hepatitis B and C, their damage comes by chronic infection of individuals, leading to loss of liver function and development, as I said, of this hepatocellular carcinoma. Hepatocellular carcinoma is a very serious liver cancer resulting in very, requirement for liver transplant or severe resection of the tumors, harsh chemotherapeutic treatments. So even now, the survival rate for 5 years is less than 10% once hepatocellular carcinoma has been developed. So to kind of stop that, vaccines for hepatitis B are now implemented as part of the infant vaccination protocols in the United States since the early 90s and throughout much of the developing world. And this has dramatically caused a decrease in the incidence of hepatitis B associated hepatocellular carcinoma in the United States, much of the Western Hemisphere, and the developing world. Healthcare workers are vaccinated for this as well, and their titer is checked every few years to make sure that they're protected. In terms of worldwide incidence, hepatitis B is still the most prevalent serum hepatitis virus, with an estimate of 240 million cases, localized primarily to sub-Saharan Africa, Western Africa, Asia, Southeast Asia, with the highest incidences. Hepatitis C is about a fourth that, about 70 million cases worldwide, again, to underdeveloped nations, sub-Saharan Africa, some of Western Africa, but the former Soviet Union and Eastern Europe are hotspots for this as well as Greenland, of all places. In terms of the hepatitis B virus, it essentially, the natural history of this virus is that it has been with humans for an estimated 40 to 50,000 years. Sequences of this virus have been identified in Aboriginal population fossils. So this virus has evolved with us. And although it was distinguished from infectious hepatitis back in 1947, the causative agent wasn't isolated until 1966, and the genome sequence wasn't determined until 1979. But identifying this agent as and isolating hepatitis B virus earned Barry Bloomberg and Daniel Gajicek the 1976 Nobel Prize for Physiology and Medicine. So although hepatitis B was identified in the 1960s, and there was a blood test developed for this even prior to the isolation of the virus, there were still a lot of transfusion-associated hepatitises that could not be associated with either hepatitis A or B viruses. So this was called non-A, non-B hepatitis, and it was called that for about 15 years. And it was suspected of being a virus because treatments that limit the replication of viruses, either with interferons or with antiviral compounds, decreased the hepatitis symptoms in patients with non-A and non-B hepatitis. So it suggested the existence of a virus in this chronic condition, even though they hadn't isolated this. The isolation and the demonstration of a virus with non-A, non-B hepatitis virus is the subject of tonight's talk, and the isolation of hepatitis C. So prior to discussing that work, I just want to refresh everyone's memory about Koch's postulates. So Robert Koch and Friedrich Loeffler established a set of principles by which you definitively associate an agent with a disease. And these postulates were published in 1890, and they have four basic premises. That one, you always find an agent with the disease. It's not found in healthy individuals. You're able to isolate that agent in pure culture. They were dealing primarily with bacterial pathogens, but isolate a viral agent or parasitic agent away from any other type of contaminating possible agent. Reintroducing that infection with that isolated agent, and then identify and recapitulating disease, and then being able to re-isolate that agent from the disease state. So I give you this kind of background because it's important to understanding the work of our Nobel laureates. A driver of this research has been Harvey J. Alter. He was born in New York City in 1935 and got his bachelor's and medical degrees at the University of Rochester. Then he started in a series of residencies and postgraduate research opportunities at Strong Memorial and then at the NIH, working with hepatitis B virus, 61 to 64, followed by a residency at the University of Washington, and finally returned to a position at Georgetown University Hospital. His academic appointments are at that Georgetown University Hospital as well as at the NIH. And since the early 1980s, he's been the chief of the infectious disease and transfusion medicine sections of the NIH. Now, his work started with Baruch Blumberg, one of the recipients of the Nobel Prize for identifying hepatitis B. But in his graduate research work or postgraduate research work, they identified a protein in the serum of patients that had what later turned out to be hepatitis B, but it was a protein that was reproducibly identified in patients with chronic hepatitis. So based on the presence of that protein, a test was developed to screen our blood supply, human blood supply. And this greatly decreased the amount of transfusion-associated transmission of hepatitis. But it didn't ablate the existence of transfusion-associated hepatitis. There were still some patients that got hepatitis even though they didn't have hepatitis A or B. And so this non-A, non-B hepatitis was noted. Serum samples were archived, and Harvey Alter was able to, with other colleagues, regenerate hepatitis by injecting serum from these patients into the livers of non-human primates. In this case, they used chimpanzees. And once they had isolated serologically a defined agent in non-A and non-B hepatitis, they used these samples to generate antibodies and went back and screened all the non-A and non-B hepatitis samples that they had, and they found that it was common to all of them. So the next important step of the fulfilling Koch's postulates came from Dr. Michael Houghton. He was born in 1949 in the UK to working class parents, but became very interested in microbiology when he read a biography of Louis Pasteur. He did his bachelor degree at the University of East Anglia and did his PhD at King's College London in 1977. He then went into pharmaceutical research for a Searle company and then went to Chiron in 1982. And while he was at Chiron, and he'd been keeping track of the non-A, non-B hepatitis virus work, he got access to samples of the serum from and plasma from the monkeys used in the Alter study with colleagues at the CDC, Drs. Chu, Kuo, and Bradley. And he isolated agents from that plasma. So they pelleted by ultracentrifugation all particles that were in that plasma, isolated DNA and RNA from those samples, and then clone those into a lambda GT expression library. So that was a phage system in which you could clone DNA fragments or reverse transcribe RNA and clone those cDNA fragments into a system where they could be expressed, the encoded proteins could be expressed. And by doing that, these proteins were then transferred to membranes and probed with antibodies from non-A, non-B hepatitis patients, human patients. And by doing so, they identified a clone that had what looked to be a full-length genome of a flavivirus, a flavy-like virus that they called hepatitis C. However, when they expressed the RNA of that virus, of that putative virus, and injected it back into monkeys, it did not generate infection. Which brings us to the third researcher and third recipient of the Nobel Prize this year, Dr. Charles Rice. He grew up in California, got his BS degree in zoology from UC Davis in '74 and his PhD in biochemistry at Caltech in 1971. But he stayed at Caltech and did a 4-year postdoc in a group that was studying RNA viruses, and his work there became very influential. He then received an academic appointment at WashU University in Missouri and went through the promotion and tenure process there, and then got a named professorship at Rockefeller University in 2001, and still has adjunct professorships to WashU and one to Cornell University as well. He's published over 400 research articles and has been the editor for Journal of Experimental Medicine, Journal of Virology, and for PLOS Pathogens. Now, while he was a postdoc, um, he was able, working in a lab on RNA viruses, to generate an infectious clone of yellow fever virus, a flavivirus. And by having that clone virus, he could delete certain sequences and generate a vaccine strain for that virus. He then established a collaboration with Stephen Feinstein. Actually, Stephen Feinstein had reached out to him once he saw the infectious clone work with yellow fever. Feinstein's group had been instrumental in the isolation and characterization of hepatitis A virus, and working together, they isolated, very much like Houghton's work, clones from the serum from infected chimpanzees. They got a number of different clones, and he noticed very strange things in the sequence, that some of them had truncations of the genome, some of them had mutations of sequences within the virus in polypurine-rich regions. So generating RNAs from a number of these clones, he infected individual chimpanzees, and a couple of those clones regenerated disease. And when he looked at those sequences, this is from the Science article where they were able to regenerate disease, he found that a bunch of these actually had deletions at the 3' end of the genome, and that the full-length sequence was much longer and had very distinct secondary structures that were required for the polymerase of the virus to make a second complementary strand, which is required for replicating the virus. He also found that the clones of Houghton had mutations in the central region as well. So how did their combined work satisfy Koch's postulates? Well, with Alter's work, he was able to replicate the condition using serum from non-A, non-B hepatitis patients, and found that agent in all of those serum samples. Between Houghton and Rice, they generated infectious clones, so essentially isolating that agent, regenerating infection in chimpanzees with that cloned agent, and then they were able to re-isolate that virus from the infected animals. So they unequivocally established that hepatitis C virus was a flavivirus, and that it was the causative agent of non-A and non-B hepatitis virus. So why is that important? Well, understanding the genome sequence and the type of virus hepatitis C is, it presents some unique targets that regulate different functions of the virus, like the entry of the virus, the replication of the virus, and the maturation and release of the virus from infected cells. So drugs have been developed that interfere with these different stages. And so hepatitis C viruses now can be adequately treated with these drugs. And so that's what makes their work so compelling and so important to understanding of another form of serum hepatitis. So thank you very much, and I'll take questions now.